Okay, let's toggle these on. Cool. We are live on. I'll just put the holding slide up just for a small level of consistency.
You can do it. I've got it here if you like. Like this. Is it on Twitter yet?
It will be.
Okay.
Cool. And now we need to do the embed.
Yeah, Twitter, great. Good.
Why didn't that work? Let me just do one thing. Source code. There we go. Cool. I've worked out a way of just, replacing the small piece of code in the full embed code. There we go. Cool. Final live stream is up.
Great. Let's do this.
Right, shall I let Jason in?
Yeah, let's do it.
Right, Jason, do we have you back in the call?
We are here. How are you?
Good.
Yes, how are you, Jason?
I'm very good. Hope
You're a little bit quiet. Is there any way you can?
No, it's not that. It's just me speaking. There's my presentation voice, and then there's my actual voice.
Okay, nice. You just forgotten to turn it on. I get you.
Yeah. By the way, I can't tell the two of you apart. I'm sorry.
Ben is close to different colors. Well, actually, Ben is a northerner and I'm a southerner. So this is the only difference, really.
All right. That sounds good to me.
Yeah. Okay, cool. Excellent. Thank you for joining us again, Jason Tetro from the Super Awesome Science Show.
Thank you.
And you're in Canada right now, but where in Canada?
I'm in Edmonton.
Okay.
So we're very close to the West Coast. We are currently in the process of getting some of that smoke that you've been hearing about from California.
The wildfires. I see. It's just making its way.
Yeah.
Problem having untidy neighbors, isn't it?
It is a bit frustrating sometimes, especially when you know what's going to happen. You kind of get a feel for what they're going to do, and there's nothing you can do to stop it. Although I have to admit, the gender reveal forest fire was new. I've never heard of that before.
No, that I think that shocked everyone. Yeah.
Yes.
So you can be as polite as you like, but you know.
I know. Please don't do that. Oh, never mind.
Cool. Well, welcome back.
Thank
I think
you.
you're the only speaker to have 2 slots on the program, which is a very special honor.
Yes, I'm very, I'm thrilled because it gives me an opportunity to sort of focus on one aspect of our relationship, and now I can focus on the other.
Cool. Well, I'm going to get out of the way so you can take us on a journey through the microbial world.
Sure. Host disabled participant. I need you to start the screen sharing for me.
We've done a brand new stream, so here you go.
Perfect. There we go. And screen and share. Oh, wait, we'll just put that there and share. And then we'll put that together. And now you should see a slideshow.
We do. Perfect.
Yes.
Excellent. Okay, well, thank you once again for having me back. And we're going to talk a little bit more now about the relationship between us and the microbes at a more concentrated, at a more intimate level. Essentially, we're going to get into something everybody's been talking about, and there have been numerous lectures on it. But before we do, let's just take a look at previously how human nature has been before today. We tend to be at war. There's no doubt about that. The thing is normally we've been at war with other humans. It's only been in the last 100 and so years that we've really been having a fight against microbes. And as I said in the previous talk, it started with them essentially spoiling our beer, and now it's essentially them spoiling our ability to go out and have fun. I mean, it's kind of a frustrating thing because as much as we might believe we can win wars, it's very difficult to do so against an enemy that is just so easily that can easily destroy us. So in that context, that latter war against microbes hasn't gone over very well. And back in the 1950s, when we first had the idea of antibiotics rampantly used, we thought we had won. Unfortunately, we were just being overconfident, and tuberculosis is a perfect example of that. And so really what's happened in the last, I'd say, 40 years is we've taken a look at germs in a different type of light, and we realized that maybe we just should not have been approaching germs from the perspective of the enemy and constantly have to destroy them. And in fact, in a way, I feel like we've lost our way. Now, that being said, I can't really blame humanity for not liking germs. I mean, after all, relationships are hard. And if you go to the self-help books, you'll find that there are numerous relationship therapy models that are out there. It's just that getting the love you want, he's just not that into you, therapeutic attachment, great for human to human. And it's funny because I've actually talked with someone who has developed a different version, attachment theory, Helen Fisher, and even she has mentioned the fact that from a human perspective, these are things that you can't really personify in the world of microbes and other invisible organisms, because essentially what you have to realize is that we're very, very different from one another. Now, back in the 1990s, we all use this terminology, men are from Mars, women are from Venus, when it comes to the idea of describing relationships between people who are very different. Well, we sort of came up with a different version of that when I was sitting with my editors way, way back when we first came up with the idea. And it sort of goes along the lines of humans are from Earth, but germs happen to be from, and you have a bit of choice here. You know, could have been Mercury, could have been Jupiter, could have been Saturn, could have been the planet formerly known as Pluto, but no, it happens to be, well, Uranus, if you happen to be an astronomy person and you want, you know, proper spelling and pronunciation. But if you do it improperly, then you pretty much get an idea of where it's coming from. Uranus. And that brings me into the basic concept of the relationship that we individually have with microbes, and that happens to be our microbiome. And so I showed this slide in the previous talk. Now what we're going to do is we're going to get into this, and we're going to learn how the microbes that are inside of us contribute to our well-being and possibly could lead to problems, usually as a result of something that we've done. I know it's really weird because when you hear about relationships, you know, oh, it's not you, dear, it's me. Germs don't say that. It's basically us. And when all of this was compiled together, it came out in the form of this book, the Germ Files. And the funny thing is that when you talk about a book that's devoted to microbes, you wouldn't necessarily expect it to do well. But we did actually make the Canadian bestseller list for several weeks. And it prompted an artist here in Canada to put this together, Germs, a love story. It ended up in one of the national newspapers. It was absolutely beautiful and brilliant. And if you actually take a look, you'll notice that there are faces on every one of the germs, and they're being very nice to us. And that's really what we're looking for. So how does the relationship work? Well, we already talked about the general human relationship with microbes, and that was the germ code. You know, the 5 ways to get into the body: ingestion, inhalation, ingress, injury, intimacy. And now we're looking inside of us. So essentially we have the microbes inside of us. Now what is going to happen? How does that relationship work? Now, when I was writing the Germ Files, I was fortunate enough to also be co-editing an academic book called the Human Microbiome Handbook. And while some of the information that is in there may now be out of date, the actual basis for a lot of what I'm going to talk about still is maintained in this one. So if you want a public version, you have the Germ Files. If you want something that's more academic, then I would really recommend that you pick up the Human Microbiome Handbook. So we go back to those 3 types of relationships. And we have the parasitism, we have the commensalism, and we have the mutualism. But on the outside, it's really easy to sort of understand from a very large perspective how these 3 work, how they're spread, how you know, plagues, pandemics, whatever can end up creating a havoc all over the world. But what about when it actually gets inside of you? What happens when a microbe gets inside of you? How do we determine if it's a parasite, a commensal, or a mutual? Well, there's one particular system that's in charge of that, and that happens to be the immune system. Now, this is abbreviated. This is only about 30%. And I must confess, I'm both a microbiology and immunology expert based on my 30 years of work. I never really got more than about 50% of the immune system figured out. There are just certain things about it that even I cannot understand, and I reach out to people. So the immune response is incredibly complicated. But what I can do for the purpose of this talk is bring it down to 4 general immune states. We have stasis, or if you wish, homeostasis, and that just essentially keeps everything in check. That's just when we're feeling good and normal. Now, if we do end up with something that we don't like inside of us, well, then we fight. And so that is the essential reaction of trying to get rid of whatever happens to be there. Now, that's parasite. And we definitely do not want that inside of us. And what ends up happening is that if we end up with a type of parasite that we don't really understand, and this doesn't necessarily have to be a microbe, it could actually be a plant, it could be a food, then we end up in overreaction. And it could be, and it leads to what's known as allergy response. And then we have one further state which happens to be the one that we're going to talk about most in the next little bit of this talk, and that happens to be something called inflammation. And the reason I want to talk about it is because it's the one thing we've heard about a lot when it comes to our internal relationship with microbes. See, inflammation essentially was thought to be at first a preparation. So it's keeping the body ready for a fight. And we have essentially an entire system, a pro-inflammatory system, that's designed to just make sure that we're ready for any kind of fight. What we've also learned, though, is that inflammation can take on a fighting all by itself, and it can also do so actually as a means for healing. So if you have an injury, it doesn't even matter if it's a cut or a stroke, after that has happened, we actually end up having an inflammatory response. And this is to be able to help heal. That's usually a good thing. However, if you start having minor, chronic, gentle types of wounds, things that you wouldn't necessarily notice, no fever, no blood, nothing that's causing any kind of symptomology, you may still end up creating an inflammatory status. And as that happens, if it becomes acute, it can become destructive. And that's the one that we hear about right now when it comes to COVID, that cytokine storm. And if you were with me the last talk, we talked about the W curve when it came to influenza and other types of new pathogens. And that cytokine storm is actually what it is causing that middle group of people from 15 or 20 to 40 to actually end up having severe problems and consequences. We can have low-level inflammation, which is acute, and this is where we get those hot flashes. We can get the fevers. A child who ends up having something they didn't really like, they're not going to have the same type of immune response as an adult. They may end up having a bit of fever, a bit of inflammation. They may end up vomiting. So in that context, inflammation can almost be a warning sign of something that's gone on that's bad. And then we have low-level chronic inflammation. Now, this is incredibly important when we talk about non-communicable or chronic diseases, because the reason we call them chronic diseases is that we are constantly having minor, small wounds happening inside of us, and the inflammatory response is there trying to heal us. The problem is the cure is worse than the disease. So if you happen to be obese, have diabetes, cardiovascular disease, autism, Alzheimer's, aging, all of these symptoms or all of these diseases that you're having essentially can lead to an inflammatory response that can make things worse. And I think when we look at it from that perspective, the fact is that we want to make sure we do not have a low-level chronic inflammation. That, unfortunately, is not easy to accomplish. I'm just going to take a drink here. So let's just look at one aspect of chronic low-level inflammation and how it can actually contribute. So in this particular case, if you happen to have gut inflammation, we now know that that can actually lead to worsening of cardiovascular disease. We even now know how that is happening. As you take in dietary intake, what ends up happening is that you digest the food, but you're not alone. You actually have all those lovely microbes that happen to be inside of you. And they're going to produce a number of different types of metabolites as they are also eating and taking care of the food. Now, when this happens, all right, those metabolites are going to have an effect on your physiology. We talk a lot about this in the Microbiome Handbook, but if those metabolites happen to be what we know as short-chain fatty acids, or SCFAs, that actually can be a benefit. It actually helps to maintain that stasis in the immune response. If, however, those metabolites are not inflammatory, but your body essentially sees them as being markers of injury, and then what ends up happening is that those metabolites actually create an inflammatory response inside of your gut inflammation. And then that in itself is going to go throughout the body and actually cause inflammatory responses in other places. Now, this type of inflammatory response is how we start seeing the worsening of cardiovascular disease. Because what is the one way of being able to protect against inflammation inside the endothelial vessels, otherwise known as the blood system? Well, you start to create plaques. You start to have atherosclerosis. And so what ends up happening is that over time, you may end up finding yourself with clogged arteries or clogged vessels, not due to anything that you've done, but because those microbial metabolites have actually kept you in an inflamed level. That's the type of thing that the microbiome is being investigated about. Now, if we move on to chronic effects of that inflammation due to the microbiome, what we find out is that every single person has what is known as an individual microbiome. And what can be done is to identify people based on their, what we call phenotype, their health factor. In this case, we've got healthy, Crohn's disease, ulcerative colitis. And then look at the microbes that happen to be inside of the gut to find out whether or not there are any links. Now, granted, there's going to be a lot of variation. And we still don't know what a normal microbiome is. So that being said, we can only look at the differences like we see here. Now, this is from 2012, but it still gives you a pretty good indication that depending on the types of microbes that happen to be inside of you, you may end up being more susceptible to particular types of chronic diseases. And now we're seeing this for other things like diabetes, for obesity, for cardiovascular disease, and even potentially for a risk of stroke. But this here is just to give you an example of what having a good microbiome versus one that is disruptive can actually do to you. And here we actually see a little bit different viewpoint of that. So if you have a healthy control and you look at the different types of bacteria, which I have at the bottom here, no need to memorize them, no need to learn them, you actually see that, yes, indeed, you could have a variety that's in there. But that variety may end up actually shrinking as a result of a disruption in the microbiome. And as you do that, you may end up having an overgrowth of bad bacteria, as a lot of people like to call them. That's the sort of the public name. What it really means is those bacteria that have sort of a bit of a parasitic component to them may actually grow and become, take up a greater percentage of the microbes than they normally would if you're a healthy control. And when that happens, that's when you start to see this inflammatory response happening both inside your gut and, of course, everywhere else in your body. So the whole idea is to try and find a way to get that microbiome back to normal. But again, since we don't know what a normal microbiome is, it makes it very difficult for us to achieve that goal of a proper, balanced relationship with our microbes. Now, if you were with me in the previous talk, you may also have heard about the idea of antibiotic resistance, especially with C. difficile. Well, one of the big problems about taking antibiotics is that they do what they're normally supposed to do, which is kill lots and lots and lots of bugs. And when you happen to be taking antibiotics into your own gut, what happens is you start losing that diversity. So here's a really interesting paper from 2010. I still use this because it's one of the best ones out there that actually shows how diversity is affected over the course of 24 months after only one dosage, one regimen of antibiotics. This is the type of thing that happens. And depending on what that teal color is, it could lead to increased inflammation. It could lead to differential metabolism. It could lead to possibly psychological problems such as depression. These are the types of things that are secondary sequelae to that chronic low-key inflammation that I was talking about. So the microbes themselves are not making you feel a certain way. That is not the right approach. The microbes are contributing to a state that you are currently experiencing that has a secondary effect of whatever it may be you're suffering from. And so whether it be that obesity, diabetes, cardiovascular disease, stroke risk, and depression, you are literally having an increased chance of that if you don't have a properly balanced microbiome. And that's really where we start seeing this whole idea of bringing in good microbes, those mutual microbes, those symbiotic microbes, to try and balance things out. Now, granted, for the last 20 or 30 years, there's only been one way that we've really seen how you can do that. Unfortunately, as you're about to see, that way has got a pretty bad reputation now. And that happens to be, whoops, I was supposed to take that out. Pardon me, probiotics. Now, back when probiotics were first introduced, they're live microorganisms which, when administered in adequate amounts, confer a health benefit on the host. This has to go through clinical trials. This has to be seen in a way that shows that over time, you have a change in how you are to a better person. Unfortunately, the majority of clinical trials do not do that. First off, they're usually not long enough. Secondly, they only focus on maybe one or two particular types of species. And more importantly, depending on what you're looking for, you may miss a whole bunch of other things. So when we start talking about probiotics, what I prefer to do is talk about what probiotics actually do. And that is they ferment. So when we talk about fermented foods, I'm sure that you're going to probably think about the beer again. And by the way, everything does come back to beer. It's not me. It just, the whole world revolves around beer. Wine is another option, but I'm sure you've heard of sauerkraut. I'm sure you've heard of kimchi, tempeh. These are all foods that essentially have been exposed to bacteria and essentially purposefully rotted to preserve them. But what we've learned over time is that those rotted vegetables, those rotted fruits, even rotted meats that are fermented, contain a lot of those very good metabolites that I talked about that can help to benefit your system, help to reduce that inflammation. Now, when we start talking about probiotics, we normally talk about lactobacillus and bifidobacterium because those are the main ones. However, we have seen probiotic style microorganisms that include worms, that include yeast, and we've even seen bacteriophages possibly being used as probiotics. The thing is, you have to realize one thing, and that is, in all of these particular types of cases, we are dealing with something that is going to lead to the production of a metabolite that is in your gut. And here's a little cartoon that gives you an idea of how that happens. So when you start having that kind of change where you have these metabolites that are being formed, you're going to essentially calm down the immune system. That's one thing. You can also produce a lot of vitamins. That's also going to help. We all know about how vitamins are necessary, and we've only recently heard, even though we've known about it for a long time, that if you happen to have a respiratory disease, lack of vitamin D could actually lead to a much more severe case. It's been like that for all sorts of different things. Apparently, we just seem to be focusing on COVID. So the bioconversions, the vitamins, you also have immune stimulation. And what this is doing is it's the bacteria themselves, by producing these types of molecules, are actually making your immune system feel more comfortable, more calm, less inflamed. And then there's a couple other things that these probiotic organisms can do. One is they can actually help in developing a barrier function so that any kind of pathogen or parasite doesn't actually touch your cells and thereby create an inflammatory response. They can compete for nutrients. So what they can do is they can reduce the chances that you're going to get overgrowth of potentially harmful bacteria. And they can also antagonize. And one of my favorites happens to be first off, let me just talk about the fact that they act as that physical barrier. I'm just giving you an example here. I'm sorry, I'm so excited about this particular one here. And what this is doing is, this here is actually showing you a zone of inhibition. In other words, if you take a lawn of bacteria and then you put in one of these particular types of bacteria, the lactobacillus or the bifidobacterium, you actually see a zone of clearing. Those lactobacillus and bifidobacterium are basically killing other bugs so that they can take over. And that's a good thing, because at the end of the day, what you're trying to do is you're trying to create some kind of diversity that's going to help you. And if lactobacillus can go in there and help to reduce the chances for negative or parasitic or pathogenic types of bacteria, that's going to be very beneficial. That's going to be very helpful. And so when you look at it from that perspective, what you have to realize is that if you can get more and more of these good germs, then there's a better likelihood that you're going to have lower levels of inflammation, and you're going to have lower levels, or at least a lower risk, of having those secondary sequelae due to the chronic low-key inflammation. So the thing is, you can get a probiotic off of the shelf, but you can't really trust it, can you? Because unless they've done the clinical trials, and there are a couple out there that do that. All right, and in the past, I've actually talked about them in the public, and I mean they do a very good job. But the majority of probiotics, especially things that are in like cookies and straws, you can't really trust them. But what you can realize is that since bacteria eat, since bacteria need food, since bacteria essentially have their own kind of diet, you too can do the same thing. And one of the things that we did a number of years ago was we were kind of joking around with this idea of a good germs diet. Wow, you can't say that more than 5 times, can you? Good germs diet. There we go. And what this is it's based on research that's been done to look at how the Lactobacillus and Bifidobacterium actually thrive. And that way, if you can get those types of nutrients into your gut, you're going to be helping to promote their existence. So just very, very basic examples. Lactobacillus loves bananas. That's a great thing. Bifidobacterium loves those complex carbohydrates. So, have some celery, have some fiber. Some certain types of bacteria, like the Bacteroidetes, they love their meats. So you want to make sure that you're having that. And what we find out is that as you're sort of going through this good germs diet, you really can find yourself, and this has actually been done in different ways when it comes to improving health. You start to see that balance in the body. You start to see that better immunity. You start to have that overall well-being. And to be honest with you, you actually start to see the relationship grow even stronger. Now, here's the thing. The reason the good germs diet actually didn't work, and I can't really put the slide up because people get upset and everything like that, but I'll tell you this. When we actually developed the good germs diet and we came up with all the different types of things that you can eat, we actually found out that there was already a diet that does that. There's already a good germs diet out there. It's called the Mediterranean diet. Now, we've gotten in trouble in the past for just trying to promote the Mediterranean diet over others. But what I will tell you is that if you look at the Mediterranean diet and you were to do the research to look at how the good bacteria that help us to maintain a balanced, low chronic low-key inflammation, you'll actually see it's the same thing. So just a little bit of advice. Now, the idea of probiotics in the human context is great, but as we know, animals also have a problem when it comes to maintaining their balance. And we've been using antibiotics in them for a very, very long time. So can we actually use probiotics or good germs to be able to reduce the chances of problems in animals and thereby reducing the use of antibiotics? And the research continues to be going on for this, but it is actually showing that, yes, indeed, if you are using the probiotics in animals, it is helping to maintain their safety. We're maintaining economic viability, particularly with the smaller farms, and we're having less of an impact on the environment. There was a study that was done numerous years ago where they actually looked at the presence of antibiotics downstream from farms and rivers, and it was, it was pretty bad. So the idea is that if we can do the same thing for us as we do for our animals, or we do the same thing for our animals as I want us to do for humans, then we are going to be in a very, very good situation. in terms of those benefits of good germs, I mean, I talked a little bit about this in the previous talk, but we have this idea of healthy supplements, the antioxidants, the fish oil or omega-3s, the vitamins, that type of thing. What if we actually were able to genetically engineer, using responsible genetic engineering, RGE, to be able to develop these, much like we have done with insulin, vaccines, and antibiotics, so that not only are we then ingesting probiotics based on that are essentially going to be beneficial to us, but then they can be super beneficial because they're also producing other things that are missing from our diet. Imagine a bacterium that could theoretically produce a vitamin D at a high level. I mean, these are the types of things that we probably will be looking at as we move forward in the future. We also have the idea of using good germs to be able to do control so that we don't have to use chemicals. Insect control definitely is an idea. We already hear about the Wolbachia bacteria that can help to reduce mosquito populations. Food safety, this is where we start seeing the work of bacteriophages. The phages are actually killing the bacteria, and it's absolutely wonderful to see how that is. Some of my colleagues are focused on this, and I'm sure that you've heard of Stephanie Strathby, who actually used bacteriophages to save her husband's life. She wrote a book about it. I really recommend that book, of course, after you've read mine. And then finally, the idea of malaria prevention. Could we use bacteria in such a way that essentially malaria still cannot thrive in the environment so that it can't get into us? These are all things that we're looking at as being possible for the future in terms of good germs. So as I'm about to finish up here, I just want to sort of revisit the relationship, both from the previous talk and also for this one. We know that there are bad germs. We know that there are good germs. You can call them bad, good, whatever you want. We can call them symbiotes and pathobionts. We can call them parasites and mutuals. It doesn't really matter. What we do know is that there are certain numbers that are good for us and can help us, and there are certain numbers that are bad for us and we want to avoid at all costs. And so when you think about it, we're not going to be able to win that war. They're not out to destroy us, but they will evolve to beat us if we try to get rid of them. If we work with our allies, we can actually work to develop not only better humans inside of us, but also better environments. And of course, a better situation for all of us when we go out in the public. I mean, imagine not worrying about getting malaria when you're in a jungle. I mean, it'd be amazing. And so in that context, what I want you to do is think about those really beneficial germs and realize that, that 1,450 pathogens that we know about, including COVID, yeah, we got to do something about them. And we know how to do that. We have our hygiene. We have our ability to avoid, all the things that you're hearing about. They're long tried and true and tested. As for the good germs, you just want to keep them getting closer to you, whether it's through your diet, whether it's through getting out there into the environment and increasing your diversity. There are always ways that you can do that. And so what I'm hoping that you can do is learn that you can actually love the microbes, especially the ones that are good for you, and give that tough love, in other words, avoidance and killing, to the ones that potentially cause you or at least threaten you in terms of your health. Now, the last thing I do want to bring up before I leave is what I call the arc. And this is how we develop our relationship, not only with humans amongst humans, but also between humans and bacteria, so that we can really close the gap between Earth and Uranus. And the first one is appreciation of our relationship with germs. And I've talked about it now twice. It's complicated, but the fact is you can read, you can learn, and you can actually not change your life a heck of a lot and have a much better relationship with them. Respect the germs, because for the longest time, we didn't respect them. We just saw them as an enemy that needed to be destroyed. And it was very, very poor of us. So let's not do that. Let's stay focused on respecting them, knowing our place with them and what they can do when we work with them versus what they can do if we either ignore them or try and get rid of them. And finally, a commitment to that relationship. I mean, relationships go through ups and downs. And right now we are going through a major down when it comes to relationship with germs because of that COVID. But you should not lose the respect, appreciation, and commitment to all the others that are out there that are essentially going to be able to help us. One germ should not change the way we look at the entirety of those species. It's just as simple as that. And so in that arc, what you can do is go and get those good germs, the fermented foods, probiotics, change your diet so that you're eating nutrients that these particular types of bacteria like. Avoid antibiotic-laden foods, avoid meats that have come from animals that were given antibiotics. Try and limit that use. Get out more, get outside of a city more, try and get more rural exposure. And more importantly, I think anything else is just realize that, when you are walking around, it's not just you. You've got trillions of microbes inside of you and at least billions that are outside of you. So think of yourself as a collective, as an ecology, as you're walking down that street, and realize that everybody around you is also an ecology. So that while from an ecological perspective, you may be conspecifics, human versus human or human with human, it also means that you are collective ecologies interacting. Let me tell you something, when you look at it from that perspective, you really understand the sense of not just our relationship with microbes, but our relationships with ourselves. You can learn more about the impact of germs in our lives. I mean, yes, you can read the germ code, the germ files, the microbiome handbook, but at the end of the day, there's lots of science journalists out there, lots of bloggers, and the academic literature. Oh my goodness, there's so much good information that you can find in there. It doesn't matter if you go to PubMed, Google Scholar, or just FEMS, because they are the leaders in making sure that we have a very good understanding of microbiology. Although I will put in a plug for Nature Microbiology because they did do a really great job of reviewing my book, and I thank them for that. And finally, promote germs for good. As much as I'm a germ evangelist, I would hope that you could be as well. So that when we start talking about individual lives, we can do something about it. When we talk to our family and friends, we can do something about it. We can also start talking in a way that will have links to policy, that will have links to politics. We know what happens if we don't involve politicians, politics, and policymakers in our decision making. We end up with well, not just probable, but actual disasters. So I think you really need to be focusing on that. And finally, whenever you see something that's very, very cool about the microbiome, and it doesn't have to be positive, it could be very negative. It could be, oh, well, look, it actually doesn't do that. Then share it. Because the more information that we have amongst our peers, the more likely it is that we're going to be able to increase the learned nature of everybody around us. Awareness is just as important as appreciation when it comes to the ARC. And I just hope that you maintain that awareness and share it with others. And when it comes to being aware of science, well, as I finish off and say goodbye, I want to just let you know that my super awesome science show is going to increase awareness about COVID. We're starting on September 28th, our second season, and you can get it anywhere you listen to your podcasts. Thank you very much.
Thank you, Jason. I'm going to tune in on the 28th and hopefully catch up with the latest developments in COVID-19 then.
Yes.
Thank you very much. So we have to bid farewell to you, Jason, but thank you so much for filling up 2 slots in the program, a noble duty for International Microorganism Day. And we're getting towards the end of the day, which doesn't ever seem like something I'd ever conceive of. We've only got about an hour and 20 minutes left, but it's been fantastic to have you, and you brought us fantastic knowledge and microbial insights. So thank you very much. Our next speaker is here. So I'm going to politely ask you to leave and bring in Carly. So yeah, just to introduce our next speaker, we have Carly Grant from the Department of Plant and Microbial Biology in the University of, from Berkeley, University of California. So it's actually quite a normal time of day for you. It's just become, well, what time is it now?
It's 2:40 now. It's a
2:40.
lovely afternoon.
Nice, lovely afternoon. And well, it's nearly midnight here, so we're approaching the end of the day. Yeah, so you've got a talk about it's on the inside that counts, exploring organelles within bacteria, which is fascinating because we're always taught there are no organelles inside bacteria in the same way as you'd expect. So I'm going to pop off the screen so that I'm not distracting you or getting in the way, and hand over to you.
All right, thank you.
There we go. We'll just check you can share your screen okay. Ah, perfect. We can see everything. Great.
Excellent. Oopsies. Oh, I forgot to start at the beginning. Sorry. All right. Hi, everyone, and happy International Microorganism Day. As you heard, my name is Carly. I'm a postdoc in Arash Kameli's lab in Berkeley, California. Please feel free to email me if you have any questions, or find me on Twitter. I'd be happy to answer them or just chat about anything. And today I'm really excited to talk to you about a fascinating area of microbiology on the topic of organelles in bacteria and also some archaea. And that may be surprising for some of you, because when we first learn in cell biology about cell plans of organisms, you might learn that prokaryotes, being bacteria and archaea, are these really simple cells, these sacks of enzymes that lack any sort of structure inside of them or organization. And they're often defined by what they lack compared to eukaryotic cells, which contain a nucleus separating their genetic material from the rest of the cell, as well as other lipid-enclosed organelles that are really important for specific functions in the cell and have really led to life as we know it today. And so while we can see eukaryotic organisms with our naked eye and how different they are, they really only represent a small portion of the tree of life, as compared to the bacteria and archaea that are really abundant and really diverse and are invisible to our naked eye. And when we start to take a closer look at some of these bacteria and archaea, you'll find that they don't all look like the textbook example of this sack of enzymes. And in fact, some of them also have these specialized, by definition, organelles that carry out specific functions and are really integral for these cells to survive in a particular niche. And here are just a handful of my favorite examples of organelles in bacteria and archaea. And each of these organisms has a different organelle that's really, really important for their lifestyle. For example, cyanobacteria, such as Synechocystis, shown here, has thylakoid membranes that house the protein complexes that are essential for the light harvesting reactions of photosynthesis. In addition, these bacteria have carboxysomes, which aren't a lipid-enclosed membrane like other organelles by definition, but they're instead made up of thousands of proteins. And inside this organelle is the important site of carbon dioxide fixation and is really essential for these organisms to survive. In addition, there are really large organelles that have been found, such as the anammoxysome in some anammox bacteria. These organelles take up about 60% of the cell volume. That's huge. And they contain the important anammox metabolism. And this metabolism creates toxic intermediates and is perhaps why this organelle is so important for the cell. In addition, this anammoxysome contains tubules of unknown function, as well as iron granules. And most unique about the anammoxysome, or another unique feature, is that in most bacteria and archaea, the ATP generation and the electron transport chain is located at the cellular membrane. However, with the anammoxysome, these processes are carried out at the anammoxysome membrane. Thus, it is maybe more analogous to the mitochondria in eukaryotic cells. And then there's an example of archaea. So Ignicoccus hospitalis, also or named after a hospitable fire sphere, because it is the host of a parasitic nanoarchaea. It's a hypothermophile found in hydrothermal vents north of Ireland at temps around 90 degrees Celsius. It is really unique, not just as being the host for a nanoarchaeon, but because it separates the genetic material from energy conservation, and thus could be a really cool example and great example model system for studying how proteins and membrane trafficking occurs outside of the eukaryotic domain of life. And finally, there are totally unknown bacteria. All they are an observation under a microscope. So here we have Ovobacter propellins, who has the cytoplasm just really concentrated in the periphery of the cell that surrounds a really large vacuole with inclusions. You'll also find in this cell box-like organelles with stacked membranes, and there's no known function for this, again, because this bacteria has only been observed. They can't grow it in the lab. In addition, this bacterium has 400 flagella per cell, and they're all found in a little or a large cavity on one side of the cell. And this bacterium is one of the fastest. It moves at 1 millimeter per second. That would be proportional to humans swimming 300 meters per second and is 100 times faster than the fastest human. And in the Comeli lab, our favorite organelle, or the main studied organelle in our lab, is that found in Magnetospirillum. And these are called magnetosomes. So they're these lipid-enclosed organelles that contain a crystal of magnetite. And they are aligned in a chain in the cell, such that the entire cell will move with the magnetic field. And it's thought that these magnetosomes are really important for the bacteria to find just the right amount of oxygen, because it's a microaerophile. It wants not too much or too little oxygen. So it's thought that it uses the magnetic field to align and search for this perfect area found in the oxic-anoxic transition zone. And we've been studying this bacteria for a long time, both the Comeli lab and also many others. And a lot has been learned about the specific proteins that are important for creating this lipid vesicle, which then is the site of magnetite biomineralization. However, there are many different and diverse magnetotactic bacteria, or these bacteria that make magnetosomes, that have been found. And these magnetosomes are beautiful. There are so many different sizes and shapes, and they're found in so many different diverse bacteria, including multicellular bacteria. And so in our lab, we're also interested in understanding how some of these other magnetite or even greigite, which is an iron sulfur crystal, how they can be formed. And for that, we turn to Desulfovibrio magneticus. And Desulfovibrio magneticus, unlike the model Magnetospirillum species, makes these tooth-like crystals. In addition, it is a strict anaerobe. And so perhaps the hypothesis for the function of magnetosomes is a little more complicated for this bacterium. And so when we try to figure out what magnetosomes are doing and understand how they're forming, we want to first ask just what are the steps of magnetite biomineralization? And this is pretty easy to do. You don't need to do genetics or anything. You simply grow the bacteria without any iron. And without any iron, they won't make the magnetite crystal, which contains iron and oxygen. And then when you spike in iron, you want to see this intermediate phase. So do the crystals form in a chain in the cell, each growing at the same time or whatever. However, when Dr. Megan Byrne, a former postdoc in the lab, performed these experiments, she found this wild intermediate phase where the cells, within 15 minutes of adding iron, filled with these electron-dense inclusions, or granules. And over time, they sort of clustered up, got bigger, and then eventually the cells would make magnetosomes. So she thought, well, are these magnetosome precursors? Because she found that these granules that filled up in the cells were amorphous, unlike the magnetite that's crystalline. They contained were bound by a lipid membrane, or just a membrane, and they contained iron, oxygen, and phosphorus. Importantly, both Meg and another former postdoc, Lila Ron Lee, found that they're likely not magnetosome precursors. And this is because the iron from these granules didn't end up in the magnetite crystals eventually. Also, they weren't sufficient for making the magnetosomes. And finally, the genes required for magnetosome formation in Desulfovibrio magneticus were not essential for the granules that formed. And so when I joined Arash's lab as a graduate student, I was really interested in understanding what these granules were, which we now call ferrosomes. So what I did is I made cells that formed the ferrosomes. I separated them from the whole cell, so I broke open the cells, removed the ferrosomes, and identified the proteins that were stuck to the ferrosomes. And with this, I found a small operon of 3 genes that were highly enriched with my ferrosomes. One of these, of the 3 genes, only one encodes a protein with a functional annotation, and that is a membrane transporting P-type ATPase. And P-type ATPases are found in all domains of life. They are, some are essential, and some, such as these P1B transporters, transport heavy metals or just metals across a membrane, which is pretty cool because we found that ferrosomes have iron in them. So maybe this is a transporter that gets iron into ferrosomes. So my work showed that, or I guess next I wanted to find what other bacteria could make ferrosomes. So I looked at who has the genes for the ferrosomes, and I found a large group of bacteria and archaea that have the potential to make ferrosomes. And I found through this work that many different bacteria that are highly diverse can make ferrosomes. I've deleted the genes and shown that they're essential for ferrosomes. And I invite you to check out our preprint for more information on this. So I feel like I've gone very fast, but hopefully what I've shown you today is that bacteria and archaea are not all like the simple textbook example. They're highly, highly complex, depending on where they live and what their lifestyle is. And rather than just calling them these simple sacks of enzymes and just simple cells, I think a lot of textbooks need to change that and say that their complexity is beyond the scope of the textbook, because they're not all E. coli and they're not all so simple. And with that, I would like to thank you all for joining me today in celebration of microorganisms, and thank you to the organizers and Joe and also Pedro for inviting me and sharing the date with me. I didn't know about International Microorganism Day before. And then I also want to thank or shout out to Small Things Considered and also This Week in Microbiology as some real inspirational sources. And with that, I would love to take any questions. Oh, good. I didn't go too fast.
Yeah, well, no, we still have a few minutes.
Perfect.
So I have a question, because I actually, at the beginning of the day, had a talk with Pedro. So is it through magnetotactic microorganisms that you know each other in terms of the research that you've done?
Yes.
Yeah, because I mean, I just can't get my head around the fact that there are these tiny magnets inside microbes and they do all these amazing things.
Yeah.
But yeah, no, so you showed us an what's it called, an organelle, where most of the bacterium was taken up by the organelle.
Mm-hmm.
So does that mean it has to perform all of its other life functions in the tiny space around the large organelle?
I think, so mostly there are unknowns with that, I would say. In the tiny space around the organelle, they think that carbon fixation is happening, as well as presumably DNA replication, transcription, translation. So yeah, you would expect it all to be packed in there. But because most of those organisms aren't, or none of them are in pure culture, and none of them have a genetic system, it's really mostly unknown, and they've made a lot of really amazing advances just without genetics, which is pretty awesome.
Okay. And then people are trying to apply genetic methods now to kind of elucidate this a bit more.
Say that again?
Are people now trying to use more genetic and genomic methods to explore the organelles?
I think in only a handful of bacteria, we have the tools for genetics, which is a big holdback, but
okay.
I would think that they're working on getting to genetics, but I'm not sure.
Okay. And yeah, my final question then was, you know how so in eukaryotic cells, things like mitochondria might have their own separate pieces of DNA. Is this the case with bacterial organelles? Are they more likely to have, you know, other pieces of DNA inside them, or what kind of examples are there out there?
Not that I know of. That's a good point. In most of these bacteria that I showed you, I noted where the DNA was, and that's because the researchers specifically found that the DNA was located in the cytoplasm and the organelles were separate. And with the case of mitochondria, for example, that's probably an ancient bacterium, right? So that's why it has DNA. That's, you know, a relic of bacteria.
Yeah, okay. So these didn't form in that same way, so we might not expect.
Yeah, probably not. Maybe some other organelles in eukaryotes have formed in similar ways, like the photosynthetic membranes could be, I guess, you know, an ancient cyanobacterium, actually, that has its own DNA too. But yeah.
Cool. Well, thank you so much. We've got some more speakers coming in for the final session. But thank you for teaching me some more new things. I've learned so much today. And yeah, enjoy the rest of your day in California. And been very happy to have you on board. And thank you for your talk.
Thanks. Enjoy your night.
We will. Thank you. Right. Let's bring in our next speaker. So this actually moves us in neatly to the last session of International Microorganism Day, which is Yeast Hour. Wonderful to be finishing on yeast. And we have a few speakers in this hour, but the next one is Miguel Teixeira from University of Lisbon, where we've had many speakers. So I think they've been pulling their weight this year in terms of filling the live stream. I'm just going to pass over to, let me just do one small thing. I'm just trying to get us back to gallery view. There we go. Okay. Let's make sure we can hear everybody. Cool. Can you hear me okay?
Yes.
Yeah.
Yes, good evening.
Good evening. And Miguel, we can hear you very clearly. Okay, great.
Yeah.
Isabel, are you going to introduce these next speakers or?
Yes. I'm here with them. So Iro Takashi is here, not here, not yet.
He is here. He is here. They're both here. Shall I let them both in and then?
He's not here.
He's just about to come. He's just entering.
Okay, nice. Because he is in the laboratory.
Oh wow.
Nunu is here. Oh, Iro.
Yeah.
Hello,
Hi.
Iro. Sorry for the evening. The early hour. I'm sorry. Okay, we are ready.
Great. Okay, I'm going to disappear and I'll let you guys get on with the talk. So we're starting with Miguel, I guess.
Yes,
so,
yes.
Yeah. If you're not talking, you can just hide behind your turned off webcam.
Okay.
But we'll enjoy
May I
some.
start?
Yeah, absolutely.
Okay. So welcome to the last session before the closing of this 24-hour marathon of microbiology. It is the East Hour. Well, the first talk is, we'll talk about the successful outcome of the convergence of microbiology and computational sciences to provide a very successful database for those working in yeasts. Well, the speaker is my colleague, Miguel Teixeira, from Instituto Superior Técnico, Universidade de Lisboa, the Institute of Bioengineering and Biosciences. Well, Miguel, please.
So let me start by sharing the screen. So can you see the presentation?
Yes.
Yeah, okay. So good evening in Portugal, good morning in Japan. It's really a privilege to be a part of this marathon of microbiology. So I really would like to start by acknowledging the invitation to be here today in this closing session on yeasts, and I'm also a big yeast fan, so it's a pleasure. So the title of my talk is Database in Microbiology, and very specifically one database, which is YISTRACT. YISTRACT was developed through a collaboration between our lab and a lab of computer scientists in the institute next door, the INESC-ID. And it was first launched by Isabel Sacré, our hostess, while I was still a PhD student, but it's a project that we've been working on for the past nearly 20 years. So it's really very nice to be able to present this work. But before we start, I would like to go back to the 90s, and to most people, if we ask what they remember from the 90s, they would probably not remember the same thing as molecular biologists. So to see what most people would think about the 90s, I Googled it, and the first image that showed up was this one. And of course, we see a lot of familiar faces: Nelson Mandela, the Spice Girls, Jurassic Park, and a little bit of science. If you notice the right down corner, we see Dolly. But for molecular biologists, the 90s actually bring us a very nice, very important memory, which is the release of the first genome sequences. And back then in the early 90s, very demanding, laborsome, expensive projects led to the release of a couple of genomes of relatively simple organisms, such as the first bacteria in 1995, and a little bit more complex, the first eukaryotes, a yeast, of course, the yeast, Saccharomyces cerevisiae. And we think about these efforts in the early 90s, which actually helped the development of the technology that nowadays enables us to get to genome sequences fairly easy. So immediately afterwards, in the early 2000s, we were able to have access to the sequence of very important model organisms, and also the first examples of each kind. So, for example, the first insect, the first plant, the first mammal, the first bird, the first fish. And nowadays, sequencing a genome is so easy that we continue to see a progression and exponential growth in the number of available genomes, as you can see in this graph. And this is due to, of course, the technological advances, which are moving really, really fast. And also, combined with that, the decrease in the cost of sequencing a genome. And just as an example, I'm showing you here what happens for the human genome. So the first human genome costed around $100 million, and at the moment, the price of sequencing a human genome is below $1,000. And if we go to yeast genomes, which is the organisms that are celebrated in this section of today, the price goes down to around $300 a genome. But what hasn't accompanied the same decrease in price was actually the computation underlying the analysis of these data. And you can see here the line of Moore's law. Of course, the decrease in computation costs there is a decrease in computation costs, but they didn't accompany the decrease in the sequencing and the cost of sequencing itself. And together with genome sequencing projects, a lot of projects came by working on transcriptomics, proteomics, interactomics, and other omics, making available such a huge amount of data that definitely required professional ways to deal with this kind of amount of data. And this is still a problem today. So, of course, there were databases before the 1990s, but they had nothing to do with the databases that came afterwards. And if we think about databases in microbiology, we are deemed to bump into giant databases that go far beyond microbiology, such as GeneBank or UniProt or KEGG. But if we think about databases in yeasts, we can see a few examples on here. So, of course, SGD for Saccharomyces genome database, it's Tween database for Candida species, Candida genome database, a couple of other examples of yeast dedicated databases, some of which actually have disappeared with time. But I would like to highlight one here, which is circling around, which is the YEASTRAC database, which I'll be discussing right now. So if we think about first what motivated us to build such a database. So we were working on transcriptional regulation. And of course, it's easy to motivate people on transcriptional regulation if we think about transcriptional regulation as the first step of control of gene expression. Of course, if we see a gene in a genome, that's absolutely meaningless unless the gene is expressed. And how much it is expressed, so how many copies of messenger RNA or proteins are being formed from that gene, that will reflect on the phenotype. And so, of course, to study transcriptional regulation is very interesting from a fundamental point of view, but also from an applied microbiology perspective. And we were working on yeasts with two perspectives. First, yeast as cell factories. And here the idea was to be able to use transcriptional regulation data to, for example, stimulate specific biochemical pathways or stimulate fermentation stress resistance regulators so as to optimize yeast as cell factories. On the opposite side of the ring, we have yeast as pathogens. And in this case, we don't want to stimulate anything. We actually want to inhibit the activity of these pathogens. And again, it's a very nice approach to be able to inhibit transcription factors or transcription factor networks because they control on their own the expression of numerous target genes that are involved in important phenotypes such as drug resistance or biofilm formation or virulence. And so, of course, transcription regulation was an issue. And before YEASTRAC, what happened was every time we needed to study a gene and wanted to know which are the transcription factors that control it, or to study a transcription factor and we wished to know what the targets were, we would need to go back to reading first tens of papers, then hundreds of papers, and it was impossible to go over and over and over again the literature. So we needed something that would make this search systematic. So that's when we contacted our colleagues working in computer science, and we've decided to build the YEASTRAC database. It took us a couple of years to finally release it in 2006, and it was based, it is based in a very simple view of transcriptional regulation, in which we have a protein, a transcription factor that recognizes a consensus sequence, which by binding to this consensus sequence in the upstream region of a gene is going to activate or repress the expression of this target gene. So what we did was, well, look for the transcription factors, the recognized consensus, and the target genes in the literature. And together with sequence data that we got from a number of different platforms and reading lots and lots of papers, we actually put together the YEASTRAC database, first focusing on Saccharomyces cerevisiae. And what we've done so far was to put together, or to gather from over 1,500 papers, more than 175,000 documented regulatory associations, 310 DNA binding sites, corresponding to 183 transcription factors. So a lot of information that is now compiled in a single database. And more than a repository, we wanted to make the database accessible and also usable to people working in yeast biology. So besides putting the data together, we wanted to develop or to design queries that would enable us to look at the data in the most profitable way. And so let me give you a couple of examples. So for example, the first thing we may want to ask is how can we, or what are the regulators of a given gene? And we can do that. And let me show 2 ways we can see the response to this question. In the first case, we're seeing the potential regulators of a given gene, in this case, CDR1. And by potential, I mean all the transcription factors that have recognition sequences in the promoter region of the gene we are studying. Undocumented, we have all the transcription factors that are known to regulate, demonstrate to regulate the target gene we are looking for. As a second example, we can also identify all the potential or documented targets of a given transcription factor. And there's several ways of looking at the resulting data. There's exhaustive tables, but also a couple of more graphical depictions. And this is an example which shows that besides having an idea of how many targets, we can also filter the targets based on what's the evidence, the underlying techniques that were used to identify these regulatory associations. A third example is how we can use the extract database and the data therein to analyze transcriptomics data. And for example, if you have a set of genes whose expression is upregulated in a given condition, what you can do is pick up that list of genes, paste it into this box, check for all transcription factors, press search, and what you get is something like this. And in this case, the answer of the database is YAP1 regulates 100% of your gene list. So if you didn't know that already, you would now suspect that probably the transcriptional response you're observing at the transcriptome-wide level is most of it being controlled by a single transcription factor, but not just by one. It's a number of transcription factors that's working together. And I'm highlighting in this data set, what the authors that published this data set did was to identify these 4 transcription factors highlighted in red here as the regulators of this number of target genes. But by looking at the information in each track, we can see that 10 other transcription factors should also be considered as regulators of this data set and may play a role in this transcriptional response as well. More recently, and that is since 2016, we've started expanding yeast track to other yeast species. So first to pathogenic yeasts, so including 4 candidate species, and more recently to non-conventional yeasts of biotechnological relevance. And you can see the names here: Z. bailey, Cloveromyces marxianus and lactis, Commegatella paffi, and Aerophila lipolytica. And we now have in yeast track 10 yeast species with all available regulatory data, and to this combination of sub-databases we call yeast track plus. And not only yeast track plus is a plus because it has more species, but it's also a plus because it enables us to look at these regulatory associations in a cross-species manner. And how is that helpful? So for example, you can try to predict regulation in one species based on what is known on regulatory associations in another species. So for example, here I'm showing you that the QDR2 gene in Candida glabrata is regulated by the PDR1 transcription factor. That's all we know. But we can look for who could be the regulators of QDR2, considering what is known for the Candida albicans QDR2. And what you see is this. And consider the regulators of the Saccharomyces cerevisiae QDR2, and what you see is this. So you can use this information, cross-species information, to predict a lot about transcription regulation in less well-studied organisms. And all yeasts are less well-studied when compared to Saccharomyces cerevisiae, so this can be really, really powerful. It can also help us to compare known regulatory associations. So for example, here we're seeing that for the same target genes in 3 different yeast species, how the regulatory networks are different from one another. And we can see this from a comparative perspective, but also from an evolutionary perspective, because we can eventually evaluate whether or not parts of the network are conserved, whereas other parts of the networks are not conserved. And we can also see this from a promoter region perspective. For example, here you can see orthologous genes in different species. So these are the same gene in different species. And we can see that in some cases, we have the same transcription factor binding sites in the promoter regions of orthologous genes in all species. But then in other cases, we have unique transcription factors that only show up in one or a couple of these species. and that may correspond to unique functions that have been acquired by these genes. So this could be also very powerful in terms of predicting how evolution has been working in these yeasts. So this almost final slide shows our users worldwide. We are always proud to show this slide, and we would like to consider that yeast track has gone viral, and we have users all over the world, and we have this perspectives to maintain the information in the database regularly updated, to expand it to other yeast species. It's going too fast. Expand it to other yeast species, and of course, if you're a yeast biologist working on one yeast species which is not being considered currently in yeast tract and would like to expand it, we can talk about it, because we have also worked on the automation of the construction of yeast tract-like databases for other yeast species just based on the genome sequence alone. So this is something that can be done in a semi-automated way at the moment. So by the end of the presentation, I hope that when you think about the 90s and the early 2000s, besides thinking about all these famous people or famous movies, you can also remember that the early 90s and early 2000s were the mark of the post-genomic era and the rise of a lot of very fascinating databases, including our own yeast tract. Let me just finish by acknowledging all the people involved. Besides myself, Isabel Sacre and Pedro Montero, we have been working on this from the start, but then a lot of people who throughout the years have contributed by curating data or developing new tools, and also funding more recently by joining the European Research Infrastructure Leaks here. So thank you for your attention.
Thank you. Thank you, Miguel. Well, as a user, this work is means a lot to all of us. Okay, so we are going to leave all the discussion at the end. We have to close to midnight because it will be the 24 hours marathon, so we cannot exorbitate the time. So we will discuss all this at the end. And since we are closing, the idea was just to have or to be able to make a toast with beers resulting from yeast fermentation. And the idea was just to have a PhD student in his first year working with yeasts and exploring yeasts to produce beer with special flavors, and obviously accomplished scientist, professor from Japan, who will be indeed the last one. Okay, so Nuno Bourbon Melo, the PhD student, please.
All right, thank you.
We need a toast, okay? We have to make
Yes.
a toast, please.
Can you hear me well enough?
Yes.
Okay. Okay, let me just share my screen then. All right, can you see it?
Yes.
Okay, great. So hello, everyone. My name is Nuno. I'm a PhD student from Instituto Superior Técnico at the University of Lisbon in Portugal. In the last couple of years, I've worked a little bit and studied a lot about how yeasts can impact the flavor and aroma not only of beer, but of other alcoholic beverages such as wine. And today I'm here to briefly talk a little bit about the role that different yeasts can have to attribute different flavors and different aromas to beer specifically. So I'd like to begin this presentation with a very brief history, a very brief historical context. Now, as most of you know, beer is an ancient beverage. It's probably as old as humanity started to collect, domesticate cereals and grains. And actually, the oldest known recipe dates back to 1800 before Christ, so almost 4,000 years ago. And it's written in the form of a poem to the Sumerian goddess of brewing and beer. And in this poem, the Sumerians describe not only the ingredients that they used to brew beer, but also the process itself. And what is interesting is that if we compare the poem back then to their deity of brewing, to the recipes that modern brewers follow today, we can see that over 4,000 years, the recipe hasn't changed all that much. So then, as now, people used water and mixed it with cereal grains to obtain a sugary syrup called the wort. They usually added some sort of ingredients. So in that poem, the Sumerians added honey. Nowadays, a very common ingredient are hops. And hops are actually flowers that have a bitter flavor, that attribute that characteristic bitterness to beer that actually makes many people dislike it. And they also have antimicrobial properties, meaning that they prevent the spoilage of beer. They prevent the growth of organisms that would otherwise, deteriorate the beer. And they have a very interesting story because in the early 19th century, when India was still part of the British Empire, obviously the British sent a lot of supplies to India, and one of those supplies was beer. However, they had a problem, which was that on the way, because it took a long time, beer got spoiled. They found, however, that if they added more hops to beer, then that beer was able to extend the entire trip to India and reach it in a state that was adequate for consumption. And it actually gave rise to a very popular beer style, the Indian Pale Ale, or the IPA, which I'm sure many of you know. And of course, while we're all here today, the microorganism that is crucial to beer making is yeast. It's this unicellular fungi that is able to transform the initial sugar-rich syrup into the beverage that we've all learned to love. So of course, 4,000 years ago, people had no idea that yeast were the main cause of this fermentation. They just knew that if they added all the ingredients together and they left them alone long enough, they would eventually get beer. So this is what we call a spontaneous fermentation. Now, over time, we've had a change in this paradigm, particularly in the 19th century, due to the work of Louis Pasteur, who found that alcoholic fermentation was carried out by yeast. And in the last few centuries, we've actually learned a lot about yeast, and so much so that nowadays, most of beer produced is not produced by spontaneous fermentation, but by controlled fermentation. And this means that brewers select very carefully the yeast that they want to use, and they add that specific yeast to the wort to then produce a beer with specific properties. And this is what we call the starter culture, the yeast that is inoculated or introduced in this controlled manner. And there are two main types of commercial beers today, the ale beers and the lager beers. Now, the ale beers are produced at a slightly higher temperature, at around 20 degrees, and the lager beers are produced at about 10 degrees. And they both use starter yeast, so they are both controlled fermentations. In the case of the ales, we use Saccharomyces cerevisiae, and in the case of the lagers, we use Saccharomyces pastorianus. Now, these are different species of yeast, but as you may have noticed, they share the same first name. This means that they are closely related. They belong to the same genus. And this is not a coincidence. In fact, there are many characteristics that make species of this family, of this genus, to be more exact in science terms, quite well adapted to the brewing environment. Now, one of those is that obviously they perform alcoholic fermentation. They are able to convert the sugars that come from the cereals into alcohol and carbon dioxide, and obviously give rise to the beer that we all know. They are very resilient and have great survival skills and are resistant to several stresses, including different temperatures and including the alcohol that they themselves produce and accumulate. They are typically able to outcompete other yeasts in the brewing environment, meaning that over time, they typically rise to the top and annihilate the competition. And of course, because they have thousands of years, they've accompanied us for thousands of years in this brewing process, they have become very well adapted to the brewing conditions and acquired some domestication traits. This means that they've acquired certain characteristics, certain skills that makes them even better at their job, as you would expect from thousands of years of practice. And obviously, they produce a beer with good flavor and aroma. They wouldn't be used otherwise. Now, hopefully by now, I've shown you how important yeast is in impacting flavor and the aroma of beer. This is because they produce a wide variety of compounds, of metabolites that affect its properties. Now, three of the most well-known ones are ethanol, which determines the alcohol percentage or the alcohol by volume, carbon dioxide, which determines carbonation, and glycerol, which determines body. However, there are many other different types of compounds that arguably, while present in lower concentrations, have an even higher impact in the properties, aromatic and flavor properties of beer, because they reach your nose, they reach your taste buds, and they're able to really determine the kind of profile you feel when you experience a different beverage. Now, of course, there are many different types of yeasts, and these yeasts produce many different types of compounds and different amounts of the same compounds, which in practice means that a beer that is produced with yeast A will in principle have very different characteristics from a beer produced with yeast B. And this means that we can explore this yeast diversity to produce a wide array of beers, from low alcohol to low calorie to beers with distinct flavors and aromas. And it's what attributes this high importance to different yeasts in the brewing sector, and what makes many researchers want to explore different kinds of yeasts to produce different kinds of beers. Now, I'd like to provide a couple of examples of yeasts that are already being explored for brewing and that actually have quite interesting potential. One of them is Total del Bruchi. It is a yeast that is found in many different environments, even in man-made environments, such as bread making. And it was actually the first non-conventional, the first non-Saccharomyces yeast introduced in the market as a starter, so the first one to be used commercially. And it produces a wide array of fruity and floral aromas that are very well suited for the production of certain specialty beers or certain types of beers, like the IPA or Saison. And then we have an old staple of brewing, which is Brettanomyces bruxellensis, which is used mainly in specialty beers, like the lambics produced in Belgium. And they are crucial to the profile of these beers because they produce these volatile phenolic compounds that attribute clove, barnyard, band-aid aromas to beer, which might seem undesirable, but are actually the soul behind that type of beer and very appreciated by many consumers worldwide. And finally, Brettanomyces bruxellensis can also be used because it consumes pretty much all the sugars present in the wort, including these dextrins, which are low molecular weight carbohydrates or sugars. And because they consume almost all of the sugars present, they can also be used to produce low-calorie beer. Now, I'm sure many of you might be thinking, why explore different kinds of yeasts that we do not know very well, instead of just genetically modifying the yeast that we actually know and that have been used for thousands of years, such as Saccharomyces cerevisiae? That is a very valid point of view. The thing is, and many researchers dedicate their research to modifying well-known yeasts such as Saccharomyces cerevisiae to make it produce different kinds of beers and beers with different aromas and flavor profiles. The thing is that legislation, especially in the developed world, is quite restrictive. And not only that, but even public perception regarding genetically modified organisms is still quite negative in many countries. And so there are many barriers toward the entrance of these GMOs especially into the food and beverage markets. Now, luckily for us, there are over 1,500 yeast species available to explore and to apply, not only in brewing, but in many other alcoholic fermentations. And obviously, many of these are pathogenic. Many of these are not interesting or have no business whatsoever participating in alcoholic fermentations, but it's still a wondrous world to explore with limitless possibilities. And obviously, I have no other way to end this presentation other than this. So have a pint, obviously always drink in moderation, but hopefully I've encouraged you to go out of your comfort zone, try different beverages with different yeasts, different ingredients, different methods. And luckily you can start right now because the next presentation is about an exotic Japanese alcoholic beverage. And so I'll leave you to it. Thank you very much.
Thank you, Nunu. And well, we are going to pass to the last speaker of this session is Dr. Hiroshi Takagi from Nara Institute of Science and Technology in Japan. Hiro, I'm very sorry for the 7 a.m. in Japan that you had to wake up extremely early. I'm sorry, but well, it's the last session, so we cannot. We are not
I think my time is.
And before giving you the word, I would like to inform those who are listening us, who are following us at the moment, that we are taking questions to the speakers. If you want, please, in the social media where you will be able to raise questions, and we will try to do the discussion, to make the discussion, and to ask the speakers at the end. So, Dr. Takashi, Please
Yeah,
go ahead.
I think my time is too early, right? Because now 7:30 in Japan. So I have already announced my presentation time will start from the 7:40, another 10 minutes later. So
No,
Yeah,
no,
we seem to have arrived.
no,
Mind
no
you-
problem, because we will have the discussion. It's better to discuss at the end. All the
oh, I
presentations.
see, I see. Okay, so can I share the screen?
Yes, you should be able to. Loading.
Okay. Can you hear me?
Yes.
Can you hear me?
Yes,
Yes.
we can hear you.
Okay, thank you. Okay, so yeah, it's too early, but I will start, okay? So my name is Hiroshi Takagi, working at the NIST, Nara Institute of Science and Technology. This is just only graduate student only, no undergraduate, okay, which is located in historical city, Nara, Japan. So I'd like to express my special thanks to Professor Isabel, who kindly invited me as not only one of the speakers in this session, but also a last speaker in IMD 2020. So today I'm greatly honored to give a talk entitled Enjoy Exotic Japanese Alcohol Beverage Brewed with My Yeast. So I think most of the countries except Australia and New Zealand are behind Japan. So I'd like to say good morning, good afternoon, good evening, depends on the nation where you are living. So my institute, NIST, Nara Institute of Science and Technology, is located in Nara, which is an inland prefecture near two big cities, Osaka and Kyoto. Its main industries are tourism, the textile, timber, and lumber. So Nara occupies an important position in the history of Japan. It is said that Japan's first state, Yamatai, was located here. So Nara is also a home of various World Heritage sites, such as Todaiji Temple and Horyuji Temple, and others, including numerous Buddhist art and architecture, classified as national treasures and important cultural asset. Also, Nara is very popular for lovely deer who are messengers of the gods. So now I'm the president of International Commission on Yeast, ICY, which is a part of the mycology and the eukaryote microbiology division of the International Union of the Microbiological Science Societies, IUMS, since back to 2016, in which I organized the ICY General Congress in Japan. At that time, I invited Isabel as one of the plenary lecture speakers. Do you remember? Yeah. So I was a research scientist of the Japanese food company, Ajinomoto, amino acid producers, for 13 years. So in back to 1995, I decided to change my career to the academic position in local university in Fukui, Japan, and moved to NIST, Nara, to the 14 years ago. Now, these staffs are working with me, and we have so many graduate students. They're including those from the Southeast Asian countries. My research background is mainly applied molecular microbiology using yeast and bacteria. That covers the basic studies in microbial science and practical application in biotechnology. So I will briefly show you once again the yeast Saccharomyces cerevisiae. So this is an important microorganism in basic science as a model for higher eukaryotes. Because the past Nobel Prize winner, Professor Yoshinori Osumi, discovered and elucidated the mechanism for autophagy, which is the fundamental processes for degrading and recycling the cellular components, proteins. Also, the yeast is a useful microbe in fermentation industry due to its extremely high ethanol productivity. So for example, I recently isolated the new yeast mutant that overproduces the isoamino acetate known as the key flavor. So by brewing with my yeast strains, the awamori, which is a traditional Japanese distilled Japanese alcohol beverage, has been successfully commercialized. So basically, I personally love the drinking any alcohol beverages. Also, my main research project is involved in the ethanol tolerance of yeast. Okay, but I am sensitivity to high ethanol concentrations of ethanol at night. Okay, so yeah, there are two types of the alcohol beverages as shown here. Non-distilled beverage and also distilled spirits. So among them, the sake and the shochu are very unique Japanese product by good collaboration with the fungus and the yeast. So sake is made from the steamed rice by multiple parallel fermentation of the fungus Aspergillus oryzae and the Saccharomyces cerevisiae, which produce the saccharification enzyme and ethanol from glucose, respectively. So recently, we successfully commercialized the new sake containing the amino acid ornithine, rich high level of ornithine, which is the physiological functions such as reducing the fatigue and improving the sleep quality. Brewed with my ornithine overproducer of sake yeast mutant. But today, I will focus on the awamori brewing. What is awamori? So one type is shochu, which awamori is a traditional distilled alcohol beverage made from the indica steamed rice in Okinawa, which is the subtropical islands in Japan. So its brewing process is quite different from that of other distilled spirits like whiskey made from the grain mash, the foods where the conversion from the starch to alcohol via sugar by malt enzyme and yeast. So during awamori brewing, the fungus Aspergillus luteinensis and yeast Saccharomyces cerevisiae are doing very good jobs for the preparing the fermented mash moromi and the producing ethanol, respectively. So after the fermentation, the alcohol is mainly distilled under atmospheric pressure and then is put in a barrel for aging that develops a rich and strong flavor. Okay, so isoamino acetate is known as the banana-like flavor and the key flavor, ginjoko in Japanese sake in yeast. Isoamino acetate is mainly converted from the intermediate alpha-keto isocaproate, which is an intermediate of leucine synthetic pathway. So in 3 steps, they're catalyzed by alpha-keto acid decarboxylase and alcohol dehydrogenase, and finally alcohol acetyltransferase. So alpha isopropyl malate synthase, IPMS, as shown here, is the key enzyme that controls the leucine synthesis because its enzymatic activity is subjected to the feedback inhibition by leucine. So to increase the isoamyl acetate contents in our body, we try to isolate yeast mutant that accumulate leucine in the cell by a conventional mutagenesis. So I will briefly show you how to construct the commercial almond yeast. For the application of the recombinant yeast, self-cloning yeast, which contains no foreign genes and or sequences except for yeast DNA, is more acceptable for consumers than the genetically modified yeast, GM yeast. According to the Japanese government guidelines, the self-cloning yeast does not have to be treated as the GM yeast because the self-cloning processes are considered to be almost the same as naturally occurring gene conversions. However, the food industry has not yet accepted self-cloning yeast. So I constructed our mori yeast strains by a conventional mutagenesis, which is a widely acceptable method of the yeast breeding for food industries and consumers. Okay, so how can we isolate the yeast cells with the leucine accumulation? It is generally said that the toxic leucine analog 555-3,4-leucine called the TFL is transported into the cells in the leucine transporters. It causes the misfolding of proteins into which it is incorporated compatibility with the leucine, and thereby the inhibits cell growth or cell death. However, the leucine accumulating cells are resistant to TFL to detoxify or to dilute the TFL concentrations in the cells. So we isolated TFL-resistant mutants derived from the wild-type awamori yeast. Okay, so this slide shows the method, how to isolate the TFL-resistant mutants. So basically, the parent cells cannot grow in the presence of the high concentration of TFL due to its toxicity. However, after introduction of the random mutagenesis into the genome of many cells with the ethyl methansylfonate EMS, the several colonies were shown up on the agar plate containing a TFL. So then such TFL-resistant mutants were collected and tested for intracellular leucine level. So among the many TFL-resistant mutants, we successfully isolated a very good mutant, 18-T55, with the increased leucine content in the cell, and found the heteroalleic mutations in the LU4 genes encoding IPMS, particularly the serine 542 phenylalanine and alanine 551 variants of IPMS. The leucine content in cells expressing the IPMS variants was higher than cells expressing the wild-type enzyme. So IPMS activity of the wild-type strain was markedly inhibited by leucine. This is the wild-type enzyme. But the mutants show about 80% activity, even in the presence of high concentration of leucine. Also, the IPMS activity were insensitive to feedback inhibition compared with the wild-type enzyme, so leading to the leucine oversynthesis. So interestingly, the homology modeling of IPMS suggested that the leucine binding was drastically inhibited in these variants due to the strict hindrance in the cavity. So we then cultured the yeast strains and measured the flavor contents from the volatile substances by gas chromatography and mass spectrometry. In this mutant, the both leucine and isoamino acetate contents were higher than the those to the parent strain compared to the parent strain. Also, in proportion to the intracellular leucine level, the yeast cells expressing the IPMS variants produced higher isoamino acetate than the cells expressing the wild type IPMS. So these results indicated that the IPMS variants that with removal of feedback inhibition by leucine causes intracellular leucine accumulation and also extracellular hyperproduction of isoamino acetate. So finally, in collaboration with the Okinawa's venture company, Biojet, the awamori brewed with my yeast mutant strains 18-T55 was successfully commercialized 4 years ago by the oldest brewery in Okinawa, Shinzato Shuzo, as the brand name, the Hyper Yeast 101. We very recently isolated a novel Saccharomyces cerevisiae strain from the hibiscus flowers. Hibiscus yeast, called hibiscus yeast, that produce the high level of ethanol and the 4-vinyl glycol, which is a precursor of vanillin, known as the key flavor of aged awamori. However, the isoamino acetate concentration observed in hibiscus yeast was lower than that observed in the conventional awamori yeast. So interestingly, by the whole genome sequences, we found that the hibiscus yeast is contiguous to the wine yeast and the different clades with the classical awamori yeast in the phylogenetic tree, suggesting that the hibiscus yeast does not share ancestry with the sake or shochu awamori original yeast strains. So to increase the isoamino acetate content of hibiscus yeast, we obtained the excellent mutant T25s with the leucine accumulation by conventional mutagenesis. In this mutant, we identified the heteroalleic mutations in the LUFO genes, encoding the glycine 516 serine variant of IPMS that was less sensitive to feedback inhibition by leucine and leading to the leucine intracellular leucine accumulation. In a laboratory scale, awamori brewed with the strain T25 showed a higher concentration of isoamino acetate than that brewed with the parent strain. So finally, this awamori was recently commercialized by the local brewers in Okinawa as the brand name Hanahana and also Sho Kamimura. I think there's such a combinational approach to yeast isolation with the whole genome analysis and metabolism-focused breeding has the potential to vary the quality of alcoholic beverages, not only awamori, but also other sake or beer and wine. Okay, so this, from now, I'm not the scientific section, but I'd like to give you many information about awamori. I'm happy to instruct you how to enjoy awamori. So many people drink awamori straight or on the rocks, water and ice, to enjoy its original taste and flavor. However, there are many ways to drink it with water. Okay, first of all, if you want to enjoy the mellow fragrance, the recommended ratio of awamori and water is 3:2 or 1:1. On the other hand, if you want to quench your thirst on hot days, a suggested ratio of awamori and water is 1:2. And also another way is to leave it in the refrigerator overnight. So doing so will mellow the taste, showing the new world to you. Okay, finally, please let me tell you how to get my awamori. unfortunately, two kinds of awamori are currently not available officially due to they are sold out. However, there are several ways to get them. At first, directly visit the local brewery in Okinawa, Shinzato Shuzo for Hyper East, and Kamiya Shudosho for Hanahana, which are located in the subtropical islands in Okinawa. Or purchase the Sho Kamimura at their website of Amazon.com. So now you can buy almost €27 or 32 US dollars, okay, by Amazon.com. Also, I will encourage you to donate 1,000 US dollar or 800 euro to our institute fund. Okay, so if you donate such kind of high money, you can get my awamori set or Japanese chopsticks. However, the most efficient way is to become a friend with me. Okay. So regarding today's topics, my former and current staff, postdoc, and the student did a great contribution. Also, I'd like to thank all of these collaborators. Finally, these works were supported by such kind of grant from Okinawa and also Japanese government. So finally, microbial products makes us happy, including foods and beverage, the commodities, and the pharmaceuticals. I love microorganisms because they do not betray us as far as we handle fairly. Thank you very much for your listening.
Thank you. Thank you very much. Very nice talk and very adequate to close the session.
Yeah.
It's a pity that we do not have your awamori here just for a toast.
Yeah, I will bring the awamori
but okay,
the
we
next
understand.
international conference.
And I would like to inform you and all our followers that this session, as well as all the 24-hour sessions, are being recorded and were recorded. So they will be available. They will be available at the International Microorganism Day and the related FAMs and associated social media. Just to tell you that it was not a problem that you start your presentation before the time, because they will be freely available and everybody will enjoy your talk and all the other talks during the day. So we have 5 minutes, as you see. It will be a very short discussion. I don't know if the three speakers would like to ask questions each other. Okay, Miguel, please.
Thank you, both of you, for your very nice talks. I'm thirsty right now, and I would like to be your friend. So, maybe a question to both of you. The two of you talked about the use or non-use of genetically modified organisms, and there's some ways to go around it, like the classical mutagenesis. Do you think there's something wrong with the law in this respect? Are we being too strict? Is it not the same to use genetically modified organisms or use classical mutagenesis? What's your thoughts on that?
Hiroshi, do you want to comment, or should I?
Yeah, you first.
Okay, so obviously, as a scientist, even if someone who's starting now, I'm a bit biased towards saying that yes, the legislation is a little bit too strict in regards to GMOs in food and beverage. At the same time, I do understand the concerns, but it's often doesn't make sense that something that is random mutagenesis would be acceptable, while something that is targeted mutation would not. For some reason, there's the dividing line in terms of public perception and even in terms of legislation that, in my opinion, it's not justified.
Yeah, probably. Yeah, today as I showed you, the hibiscus yeast, there was also the mutagenized with the EMS. So, but recently, as you know well, the whole genome sequence technology are well developed. So it is not so difficult to get the whole genome sequence, not on to find out not only the genes which contribute to the favorite purposes, but also other favorable or non-favorable mutations become clear. So as one of the sales talk, we can give many information to the consumers, even though there's so many mutations in the genome of the new yeast mutant or strains. Yeah. So still, I think still the conventional mutagenesis is very powerful approach by the combination with the whole genome sequence or metabolism, metabolic mix analysis. Okay? But basically, as in Japan, also as the European country, also still particularly food industry and consumers do not accept the GMO and the even the self-cloning yeast. But we are discussing the how safe their genome editing.
你早上太来了以后，太早来了以后太疲劳了。昨天休息好了吗？
We're about to close the session. Well, we are closing the session. At this moment, I would like to give the word to Joseph from the FEMS office. But before, I would like to thank and to congratulate FEMS and all the speakers for this perfect marathon of microbiology. It was a privilege for me, and I'm pretty sure that for all of us that were here in this platform to interact with so nice people from the office. Thank you very much, Joseph. Thanks, Ben. Thanks to all the FEMS office and the FEMS direction. And obviously, thank you, all the speakers of today. And I will pass the word to Joseph. He has nice things to tell us for sure.
Yeah.
Thank you, Joe. Thank you, Joe.
Obviously, we've been broadcasting for 24 hours, so we can do a little bit of a before and after comparison of a few little stats just to tidy up at the end. So what we've noticed is across the IMD, Twitter and Instagram, we've seen like huge growth in the last 24 hours, and you kind of see these massive peaks. There's tons of people have started to follow International Microorganism Day who hadn't before, and so many of those have been within the last 24 hours. So that's been great fun. Now we can do a little bit of a comparison. So this was how many impressions and posts the hashtag had last night, 24 hours ago, almost to the moment. We had 3.5 million impressions, which means that something with International Microorganism Day in a post had moved over somebody's screen 3.5 million times in the last 3 months. Wow, over the last 24 hours, that's grown quite a lot, and we're now at over 10 million impressions. So we've had the same as 3 times as much content moving across people's screens in the last 24 hours as we did in the last 3 months. So that's been great. Thousands of users, thousands of posts, millions of people reached. Yeah, so yeah, nearly 4 million reached, and then 10.5 million impressions. Website's seen some great growth as well. We were kind of around 5,000 visits and 12,000 page views, and that's kind of nearly doubled, about 9,000 visits and 18,000 page views. And again, it's all happened in the last 24 hours. And then this was the geographical reach of visitors on last night. At this point, Japan was the nation with the most visitors. And in a fitting ending, I can announce the country that now has visited the website most has been Portugal. So we have just pipped Japan into lead place in the last 24 hours. But yeah, strong country showing from Portugal, Japan, the UK, US, India, Netherlands, Brazil, Italy, Spain, Germany being the top 4 countries. I think that's it from me. Other than to say thank you all very much. This has been wonderful. I have learned so much. I've been awake for a very long time, as have Ben, but it's been thoroughly enjoyable. And
well done.
My gratitude to every one of the speakers and everyone who's come and joined us today. We've been literally around the whole world in one revolution of the planet, and we've seen so many faces in so many countries, and it's been really good fun. Yeah, and it will all be available online. It is actually already available online, so if people are watching the live stream, they can always skip back to any point. So if anyone did tune in at 7:40 for Hiroshi's talk, they could always go and 7 minutes earlier and see it straight away. So there's no need to wait. But we will get the videos individually put on YouTube so that you can see them with speaker titles and everything. But yeah, thank you very much, everybody.
Thank you very much.
Thank you.
So,
and well done.
Thank you.
Congratulations.
Yeah.
Bye-bye.
Bye.
See you next year.
See you next year, everyone.
Good
Thank you
night.
very much.
Good night, guys. Yeah.
Have a great day, Hiroshi.
Yes,
Good morning.
the same. Well, you get the rest.
Are you from the
bye.
Portugal or the Portugal?
No.
I'm in the Netherlands, Ben's in Sweden. I think the rest of these guys are in Portugal.
But
Claudia is from
the
Portugal.
midnight,
Yeah,
Midnight.
we are from
Yes,
Portugal.
and Miguel Teixeira.
Good sleep.
They are from the FEMS office, located in Netherlands.
Yeah.
In Delft.
Thank you very much.
Delft, isn't it?
Well, Ben is in Sweden, right?
Yeah.
Yeah.
And you are in Sweden?
Yeah.
Sorry.
One of our remote workers.
Okay, please keep in touch.
Bye.
Okay.
I will.
Bye-bye.
Everyone, thank you.
Thank you.
It was a great day.
Thank you, Joe. Thank you, Ben.
Thank you.
Thank you.
Right. Bye.